95% of your serotonin is made in your gut, not your brain. That one fact rewrites everything you know about mood. Your brain runs on a separate serotonin system cut off from the gut. Two factories, same molecule, zero communication, or so scientists thought. Over the next 20 minutes, you'll see serotonin do things your doctor never explained. From gut bacteria to brain wiring. Level 1 starts now. This molecule controls your mood, sleep, hunger, and ability to feel satisfied, and your risk of anxiety and depression. It's called serotonin. Your gut holds 95% of it. Blood platelets carry 4%. Your brain, the part everyone talks about, has just 1 to 2%. If serotonin is your happiness chemical, and most of it lives in your gut, then gut health matters more than anyone has told you. Your brain makes its own serotonin from a cluster of neurons in your brainstem called the raphe nuclei. Those neurons reach nearly every part of your brain. One tiny cluster, billions of connections. The raphe nuclei control your prefrontal cortex, amygdala, and hippocampus. When they misfire, everything goes off. How does gut serotonin talk to your brain if it can't cross the barrier? That's level 2, and the answer rewrites everything. It starts with tryptophan, an amino acid from food, turkey, eggs, pumpkin seeds. It reaches your intestinal lining where a specific enzyme grabs it. That enzyme is TPH1, tryptophan hydroxylase 1. It converts tryptophan to 5-HTP, raw material stamped into a half-finished part. A second enzyme, AADC, finishes the job. 5-HTP becomes serotonin, full name 5-hydroxytryptamine. Your gut just made your mood molecule. Your intestinal lining contains millions of Enterochromaffin cells. They sit right at the surface, sensing everything that passes through your gut. Food pushes through, these cells feel the pressure and release serotonin. So does your stomach's pH. So do the chemicals your gut bacteria produce. That serotonin goes 2 places, into your bloodstream where platelets carry it for blood clotting, and onto nerve endings that move food along your gut. Your gut bacteria produce compounds called short-chain fatty acids, SCFAs. These signal your EC cells to keep making serotonin. Butyrate is the key SCFA. Your bacteria make it by fermenting dietary fiber. It docks on EC cells and turns up serotonin production. No fiber, less serotonin. Lactobacillus and bifidobacterium are serotonin activators. Germ-free mice raised with zero gut bacteria had 60% less serotonin than normal mice. The blood-brain barrier is your brain's security system. It blocks almost everything, including gut serotonin. Every molecule your gut makes stays outside your brain, forever. Your brain makes its own serotonin using TPH2, a different version of the gut enzyme. It lives deep in your brainstem. In your brainstem, the raphe nuclei manufacture brain serotonin from scratch. 2 separate factories, same molecule, completely different parts of your body. The vagus nerve is your gut's hotline to your brain. About 80% of its traffic flows gut to brain. Your gut is talking to your brain constantly. EC cells release serotonin onto vagal nerve terminals. The nerve fires the signal to your brainstem in milliseconds. Not blood chemistry, a direct nerve call. When your gut signals the brain adjusts its own serotonin. Feed your gut, well, brain chemistry shifts. Starve your microbiome, the signal weakens. Level 1 complete. Serotonin doesn't hit one switch. It has at least 14 receptor types, each doing something different. Same molecule, different doors, completely different effects. The 5-HT1A receptor is the brake. It slows neurons, reduces anxiety, quiets fear. Most antidepressants activate it. Buspirone, the anxiety drug, targets it directly. The 5-HT2A receptor is the accelerator. Neurons fire hard, driving perception and creativity. It's the receptor psychedelics hijack, but we'll get to that. The 5-HT3 receptor in your gut wall triggers nausea signals up the vagus nerve. GLP-1 drugs like Ozempic increase gut serotonin activity. That's why the nausea is so common. The 5-HT2C receptor controls appetite. Serotonin in the hypothalamus drops your hunger signal, which is exactly why a tryptophan-rich meal genuinely reduces hunger. One molecule, 14 receptors. Serotonin runs different programs in your gut, brain, blood vessels, and bones simultaneously. This is not a happiness switch, it's an operating system. Dopamine is the wanting molecule. It drives craving and pursuit. Serotonin is the having molecule. It signals satisfaction and contentment. They are emotional opposites. These two systems actively balance each other. Serotonin via 5-HT2C suppresses dopamine in the reward circuit. Deep contentment and wild craving cannot coexist. When serotonin is high, dopamine chasing slows. You feel full, at ease, less reactive. When serotonin drops, dopamine takes over. Craving surges. Impulsivity spikes. After serotonin does its job, a transporter called SERT recycles it back into the original neuron within milliseconds, one of the most elegant systems in your nervous system. SERT is why SSRIs work. Selective serotonin reuptake inhibitors block SERT. Serotonin can't be recycled. It builds in the synapse and receptor activation increases. The synapse floods with serotonin within hours of the first SSRI dose, but mood doesn't change for 4 to 6 weeks. So what is the SSRI actually doing? A 2022 review in Molecular Psychiatry analyzed 17 studies. Conclusion: no consistent evidence that depression is caused by low serotonin. The chemical imbalance theory went too far. SSRIs flood the synapse first. Excess serotonin triggers a chain. Serotonin receptor, then CAMP, then a protein called CREB. CREB is the master switch for neuroplasticity. CREB activates a gene called BDNF, your brain's fertilizer. It grows new connections and repairs damaged synapses. That process takes 4 to 6 weeks to build up. When SSRIs flood synapses, they hit all receptors, not just depression-related ones. The 5-HT2A receptors that drive joy and passion get suppressed too, and the result has a name: emotional blunting. A 2022 survey found 40 to 60% of SSRI users reported not depressed, but also not excited or moved. You cry less, you laugh less, just flat. The blunting is real and documented, but by week 6, BDNF rises 20 to 40%. New synaptic connections form. In some patients, that rewiring is the difference between recovery and relapse. BDNF doesn't only come from SSRIs. 30 minutes of aerobic exercise raises hippocampal BDNF 20 to 40%, the same range as a standard antidepressant, after 6 weeks. Exercise builds serotonin too. It fires serotonin neurons, raises brain tryptophan, and upregulates receptors. Same outcome as an antidepressant without the blunting. Morning sunlight does the same. Your eyes have photoreceptors connected directly to the RAF nuclei. 20 minutes of morning light turns up serotonin synthesis. Mechanism, not platitude. Your gut microbiome contains 38 trillion bacteria. At least 40 species directly influence serotonin production. Lactobacillus and bifidobacterium are the main drivers. The mechanism: bacteria ferment dietary fiber into SCFAs like butyrate. Butyrate docks on EC cell receptors and switches on serotonin production. No fiber, less serotonin. This is the gut-brain axis. Bacteria produce SCFAs, triggering EC serotonin, signaling the vagus nerve, adjusting your brain chemistry, a complete chain running with every meal. A broad-spectrum antibiotic clears your gut microbiome without discrimination. After one course, lactobacillus and bifidobacterium can take 6 months to recover, sometimes 2 years. Without serotonin-producing bacteria, EC cell output drops and the vagal signal weakens. This is one reason people report depression and brain fog after a course of antibiotics. Ultra-processed food does it slowly. No fiber means no butyrate, no serotonin trigger. Your microbiome starves on refined carbs and takes your gut serotonin down with it. Lactobacillus rhamnosus and Bifidobacterium longum have the most evidence. A 2021 review found these strains consistently raise serotonin metabolite levels, but only while you take them. Fermented foods, yogurt, kefir, kimchi, sauerkraut deliver live bacteria more bioavailable than capsules. A 2021 Stanford trial found high fermented diets raised microbiome diversity in 10 weeks. The gut-brain axis responds to what you feed it week by week. Fiber, fermented foods, fewer antibiotics. These are the direct inputs to your gut serotonin factory. Level 5 complete. Psilocybin converts to psilocin in your body. Psilocin fits serotonin's 5-HT2A receptor nearly perfectly and activates it more powerfully than serotonin itself. Powerful 5-HT2A activation cascades through your prefrontal cortex. Normal firing patterns break down. Neurons that rarely connect suddenly can. A key brain network gets disrupted. The default mode network runs your inner monologue and sense of self. Depression is partly a stuck DMN. Psilocybin disrupts it within minutes, and the effect outlasts the drug by weeks. A 2023 Phase 2 trial at Johns Hopkins found a 40% remission rate in treatment-resistant depression. Effects lasted 6 months from one session. No other treatment comes close. Psilocybin increases synaptogenesis, new synaptic connections. At 24 hours post-dose, dendritic spine density was 10% higher than baseline. More structural change than SSRIs produce in 6 weeks. LSD and DMT work through the same mechanism. All three are 5-HT2A agonists. All three disrupt the DMN and produce neuroplasticity that outlasts the drug. Let's recap. Your gut makes 95% of your serotonin. Your brain makes its own. 14 receptors run different programs. SSRIs work through neuroplasticity, not serotonin levels. Gut bacteria drive gut serotonin. Psychedelics reset stuck patterns through 5-HT2A, and one drug combination can push the system to a dangerous threshold. 4 evidence-based inputs move the actual dials: morning light, exercise, tryptophan timing, and gut diversity. Everything else is downstream of these 4. Serotonin syndrome happens when too many receptors activate at once. Combining an SSRI with an MAOI is the main trigger. MAOIs block breakdown. SSRIs block reuptake. Serotonin surges. Symptoms escalate in 3 stages: mild, tremors, rapid heart rate, dilated pupils, moderate, high blood pressure, fever, severe, temperatures above 41 degrees Celsius, seizures, organ failure, onset within 6 hours. SSRIs plus MAOIs is the most dangerous pair, but tramadol, linezolid, and St. John's wort all carry risk. If you're on an SSRI, know what you cannot combine with it. Your retina contains photoreceptors called iPRGCs that detect morning light and signal directly to your Raphe nuclei, upregulating TPH2, the brain enzyme that makes serotonin. Exercise boosts serotonin 2 ways. It reduces the kynurenine pathway that steals tryptophan and drives tryptophan across the blood-brain barrier faster. You're rerouting supply chains. Tryptophan needs carbs to reach your brain. Carbs trigger insulin, which clears competing amino acids from the transporter. Tryptophan wins the race. Serotonin production rises. An 8-week mindfulness study found increased 5-HT1A receptor binding in the prefrontal cortex. More receptors means more sensitivity to the serotonin you already have. Meditation also quiets the default mode network, the same network disrupted by psychedelics. Different mechanism. Sustained 5-HT1A upregulation, same destination. Gut diversity may be the strongest lever. More bacterial species means more SCFA production. A 2023 meta-analysis found diet diversity was the top predictor of microbiome health. Aim for 30 plants per week. Pillar 1, morning light. Outside within 30 minutes of waking, no sunglasses. Blue spectrum light activates your iPRGC photoreceptors and turns on serotonin synthesis. Pillar 2, zone 2 cardio. 20 to 30 minutes at conversational pace, 3 or more days a week. Below this dose, the effects are only partial. Pillar 3, tryptophan with carbs, turkey or eggs combined with whole grains. Evening timing maximizes the serotonin to melatonin conversion. The conversion runs strongest at night. 5-HTP can raise brain serotonin, but most converts in your gut before crossing the blood-brain barrier. The yield is inconsistent, and the evidence for mood is limited. Pillar 4, feed your microbiome. Prebiotic fiber inulin and resistant starch feeds your serotonin-activating bacteria. Eat garlic, onions, leeks, green bananas, not pills. Food. The 3-0 plant foods target comes from the American Gut Project 10,000 people. Eating 3-0 plus plant species drove higher microbiome diversity, more SCFAs, and more gut serotonin. Your pineal gland converts serotonin to melatonin in darkness. The enzyme RNAT is suppressed by light. Your daytime serotonin is the raw material for your nighttime melatonin. Serotonin and melatonin are 2 halves of a 2-4-hour cycle. Low serotonin during the day means less melatonin raw material at night. The 2 systems are one continuous loop. Blue light at night tells your pineal it's still daytime. RNAT shuts off. Serotonin doesn't convert. 1 hour of screens after dark delays melatonin by up to 90 minutes. Your bones have serotonin receptors. Gut-derived serotonin slows the osteoblasts that build bone density. Gut conditions that affect serotonin are linked to lower bone density. Your heart has receptors that regulate heart rate and vessel tone. Low platelet serotonin links to heart disease. Serotonin syndrome triggers dangerous arrhythmias. This molecule touches every organ. 95% in your gut, 4% in blood, 1% in your brain. That 1% drives your mood, focus, sleep, and impulse control. The smallest fraction runs the most complex program. Alcohol temporarily floods serotonin receptors, but chronic use downregulates TPH2. Regular drinkers have lower baseline serotonin between drinks. The mood lift borrows from tomorrow. Chronic stress activates IDO, which diverts tryptophan away from serotonin. It feeds the kynurenine pathway instead, producing quinolinic acid, a neurotoxin tied to depression. Ultra-processed food changes your microbiome within 72 hours. A 2021 study showed measurable drops in lactobacillus and bifidobacterium in just 3 days, enough to cut gut serotonin output. Psychobiotics are specific bacterial strains like Lactobacillus rhamnosus JB, one that reduce anxiety in human trials, not by changing brain chemistry directly, by changing what the gut sends up the vagus nerve. Researchers are developing gut-restricted serotonin modulators, drugs that change gut serotonin signaling without crossing the blood-brain barrier. No cognitive blunting. Phase 1 trials are ongoing. Psilocybin just entered Phase 3 trials. The FDA granted it breakthrough designation for treatment-resistant depression. If Phase 3 matches Phase 2, approval could come as early as 2027. Serotonin existed before brains evolved. C. elegans, a worm with just 302 neurons, uses it to signal food availability. Your gut serotonin system may predate your nervous system. Every vertebrate uses serotonin: fish, frogs, birds, mammals. Same receptor families, same SERT transporter, same synthesis pathway. It is one of the most conserved signaling systems in all of life. The molecule in your gut right now is functionally identical to one in a 700 million year old ancestor. It predates consciousness, and yet it runs your mood, sleep, gut, and bones. 6 weeks of the 4 pillars produces measurable changes. BDNF rises 20 to 40%, dendritic spine density increases, vagal tone improves, not feelings data on a lab report. The protocol requires no prescription. 4 consistent inputs into a system that responds predictably, not instantly, but measurably and consistently the way biology always responds. Your gut-brain serotonin axis has been working without your input your entire life. Now that you understand it, you can stop working against it and start working with it. The EC cell in this shot has been making serotonin since before you started watching. It will keep making it after. Every meal changes what it produces. You are 38 trillion bacteria and 37 trillion human cells cooperating to run one serotonin system. That system shapes how you feel, what you want, and when you sleep. Now you know where serotonin is made, how it signals, what damages it, and what restores it. That knowledge doesn't change your biology, but it changes every decision that does. Pillar 1, morning light 30 minutes outside. Before 9 a.m., no sunglasses. Pillar 2, zone 2 cardio, 20 to 30 minutes, at least 3 days a week. Both boost BDNF and serotonin. Pillar 3, tryptophan-rich food with complex carbs, turkey, eggs, whole grains, evening timing. Pillar 4, 30 different plant foods per week, not servings, different species. 4 pillars, 0 prescriptions, measurable changes in 6 to 8 weeks. Every mechanism is from this video. You've seen the biology. These are the levers. Right now, someone's mood is the result of what they ate yesterday, the light they got this morning, and the bacteria they've been feeding for months. The mechanism runs regardless. Their gut bacteria, their blood-brain barrier, their 14 receptors, their SERT transporter cycling every millisecond, their melatonin building from this afternoon's serotonin, all running quietly. The cycle doesn't stop. Your gut is making serotonin right now. The 4 pillars are either running or they're not. Now you know the difference. GABA is your brain's primary break gamma-aminobutyric acid. Where serotonin moderates, GABA actively quiets. Anxiety is often as much about GABA failure as serotonin. This channel covers every system: dopamine, serotonin, GABA, glutamate, norepinephrine, and more. The level of detail that changes how you see your own biology. The science behind what's changing you. Subscribe, and we'll see you in the next one. You saw serotonin built in the gut, blocked from the brain, routed through the vagus nerve. 14 receptors running different programs. SSRIs working through neuroplasticity, not serotonin levels. You learned how bacteria drive gut serotonin, how psychedelics reset the DMN, how serotonin syndrome is a real threshold, and 4 inputs that move the actual system. That EC cell from the first frame is still making serotonin right now, responding to what you ate today, signaling your brain. It was doing this before you started watching. The gap between knowing and changing comes down to 4 things: morning light, movement, tryptophan timing, and 30 plants a week. Inputs to a system that responds predictably. Get those inputs right, and the system changes physically. New synaptic connections form. Receptor sensitivity increases. Vagal tone improves. Measurable changes in weeks, not years. You have more influence over your serotonin than any drug company wants you to believe. Gut, light, movement, food, these are the primary inputs. You are already running this system. Right now, someone's gut bacteria are producing SCFAs. Their EC cells are releasing serotonin. Their vagus nerve is carrying that signal upward. Their Raphi nuclei are tuning their brain. Their body ran this system before the research existed, as it has for millions of years. You don't need to understand the mechanism to benefit, but understanding it changes what you choose. Your serotonin system isn't waiting for a pill, it's waiting for the right inputs. Morning light, movement, a diverse gut, tryptophan timed with carbs. The next video covers GABA, your brain's primary calming neurotransmitter. Serotonin and GABA work together to regulate anxiety. Understanding both reveals the full mood regulation system. Healthicore goes deeper than the 10-minute summary. Each video in this series builds on the last. Dopamine, serotonin, GABA, these systems don't work in isolation. If you made it this far, you understand serotonin at a level most people never reach. You have a protocol you can start tomorrow. Morning light first. Subscribe for what's next.